All right, everybody, welcome back. In this video, we are looking at Unit 7, Lesson 1, Exploration 3. This is the last exploration from Lesson 1 where we've been looking at meiosis. And in this video, we are specifically going to focus on how this process of meiosis creates the genetic variation that we need in order to have a healthy population of individuals. All right, so let's get right into it. Again, genetic variation is super important. We'll talk about this later when we talk about ecology, but genetic variation simply talks about the differences in genetic material of the individuals of a population. Now, there are certain parts of our DNA that are going to be relatively standard for all of us that makes us the species that we are, but the details of some of those physical features can be very different from each other, and that's a good thing because it helps keep our population healthy. Now, there are basically two ways, two steps in this process that impact the genetic diversity of the gametes that are produced. Remember, in meiosis, we are trying to create those haploid gametes that have half of the copy of genetic material and are designed to combine with another gamete that also only has half of the information. All right. Now, the first one of these is something called independent assortment. All right. If you remember back to our previous explorations, you have homologous chromosomes that are the same size and shape that are going to line up in the middle of the cell during metaphase 1. And during metaphase 1, they are going to then be pulled to opposite sides during that next step, which is that anaphase step. Now, again, in our previous exploration, we talked about how the whole purpose of meiosis 1 was to separate those homologous chromosomes, and this process happens randomly. The direction in which the homologous chromosomes separate is different every single time, or I should say is random every single time, and therefore is different almost every single time. It would be highly unlikely for the same exact set of chromosomes to be separated in the same exact way 2 times in a row. All right. And so this is called independent assortment, and it provides us with our first example of genetic variation, because sometimes the chromosome from your dad gets added to the gamete, sometimes the chromosome from your mom gets added to the gamete, but the combination is different every single time. Okay. Here's kind of a look at that. If this is our starting cell, it looks like we have a white, a red, a blue, and a green. When these two gametes separate from each other, we're going to need to have a large and a small on each side, but there's 2 different possibilities for how they could separate. And actually there's 4 different possibilities, but right here we could have a large white and a small blue. And then on the other side, a large red and a large green. So that's one way they could go. We could flip it. It could be a large red and a small blue, large white, small green. Also a possibility. It just depends on how these 2 homologous pairs choose to separate. And again, this process is random, and so it creates different outcomes. Now, in this cell, we only are showing 2 pairs of chromosomes. If you remember, humans have 23 pairs of chromosomes. And so the likelihood that all 23 of those chromosomes would randomly separate the exact same way is extremely low. In fact, the likelihood of that happening is about 1 in every 8,400,000 possibilities. So very, very unlikely that any of these circumstances are going to be exactly the same. And even if they are, it's very, very infrequent. Now, independent assortment provides us with one of the ways that we create randomness and create diversity in our gametes. But the second one is even more important. The second way that we create randomness is through something called crossing over. Now, remember, as we've said, homologous chromosomes are those chromosomes that have the same size and shape. And by extension, they have the same genes in the same locations. Now, at the beginning of this process of meiosis 1, your homologous chromosomes are going to form, and they're going to come and be very close together. They're going to form a structure called a tetrad. Not sure if this textbook talks about that or not, but The tetrad structure is these two duplicated homologous chromosomes that are in close proximity to each other. And while they are close to each other, they are able to trade segments of DNA between them. Any segment of DNA that codes for the same type of information can be traded between those homologous chromosomes when they are close to each other. Now this video kind of shows what that looks like. So I'm going to play it real quick for you because it's only about a minute long. As you can see here, we have our two chromosomes, a red chromosome and a blue chromosome. And right now they haven't condensed yet. Right now they're in their loose chromatin form. But as we begin this process, they're going to condense. And then as you can see here, they're going to trade comparable sections. All right, so over here, let's just say this is our chromosome from our female parent. This is our chromosome from our male parent. But it has a small section of DNA from that other parent. And remember, these are duplicated sister chromatids, so there's actually 4 total chromosomes here. When this process continues on and we separate through our second round of meiosis, what we're going to see is you have 4 different gametes that each have 4 unique combinations of DNA, all red, all blue, red with a little bit of blue, blue with a little bit of red. And so that random trading of sections of the DNA creates another layer of variation, so that even if you had 2 out of 1.84 million combinations that turned out to be exactly the same through independent assortment, the likelihood that they would have also traded exactly the same segments of DNA during crossing over is exceptionally low. And this is how we can make sure that even siblings that are built from the same gene pool do not have identical DNA structures. All right. Now this last section right here is going to talk about fertilization. Once we have those mature gametes that are ready to reproduce, the sperm and the egg cells are going to fuse and they are going to produce what's called a zygote that contains one complete set of DNA. Now this is a pretty cool process, and we talked a little bit before in the previous video about gametogenesis, about how egg cells are very different from sperm cells. But this is a picture of that difference. Look how massive the egg cell is compared to the sperm cell. Now the egg cell also has a pretty cool feature, which is that as soon as one sperm successfully fertilizes the egg and combines genetic material, the egg initiates kind of like a lockdown procedure where it releases just this chemical that kills all the sperm, shuts down every opportunity, and basically like blows up anything that's not connected already. And so all the sperm except for the one that was able to fertilize get knocked off and aren't able to continue trying to fertilize the egg. And that ensures that we only have one full set of DNA and not, 1.5 or more. So, all right, that's the end of this exploration. And it brings us to the end of lesson 1. At this point, you should be ready to take the quiz.